We're starting the second segment following these comprehensive talks in the morning. And the reason, and what we're tasked to do is to identify mature areas, low-hanging fruit kind of areas of study in the auspices of MSGERC study group, what are the unmet needs, how a modern study will look like, and everything. So the panel consists of people with good understanding of the disease and different perspectives, and my co-moderator, Dr. Miseli, and myself will just basically have the conversation flowing. And I'm going to start with a question, and hopefully everybody can basically interrupt and give his perspective. But listening to Tom Patterson talk, I was wondering if you had one study to do and all these antifungals you described, how you would do it, how you would prioritize all these new drugs, and how the next phase of an Aspergillus trial looks like to you. I know it's a loaded question. I know it's a very difficult thing to do, but just to have the conversation going. Yes.
It is a loaded question. There's no way I'm going to take the bait for that. But what I would say is kind of like the discussion happened yesterday with Canada and crypto. I think that to say we should put all our eggs in the one basket would really be a mistake. That's kind of where I would come from it. Mm-hmm. And I think, the trials that I spoke about, obviously I didn't go into a lot of details, but I think having sort of clever designs of smaller trials most of them are phase 2, so they're not very big. I think a more reasonable strategy and one that's more realistic in terms of funding by these companies, and I think more realistic in terms of the investigators being able to do it. I think that what we really need to do as a MSGERC study group and with these companies is really work hard to maximize our endpoints so we can take advantage of smaller numbers. I think if you look at that Merck trial, the POSA-VORI trial, a 600 patient trial for 5+ years, it's just not going to happen for these drugs. But I think they're all going to be high enough risk, frankly. Those people that are doing it know that, but they don't want to hear that really probably. But it is high enough risk that, frankly, not all of them are going to make it. And there's going to be some bumps in the road. I'd love it if they all did, because I think there are enough areas that we will be able to have an impact. But I think that they are doable. I think reaching some newer populations, maybe like ICU patients, is something that we can also think about. And I think if we get more diagnostics, like molecular tests, to pick up more patients, and get agencies to agree that that's a reasonable strategy to allow patients to get enrolled, because even though I talked about susceptibility, of course, most of these patients don't have a culture. So if you can have, point of use test even, Emmy has a new test for point of use diagnostics, sort of bedside. That would be a way you could pick up an early case. Yeah, it's obviously going to help our patients, but it definitely would help our trial enrollment.
Tom?
I think we have to recognize that we have several groups of high-risk patients potentially, and though the premise of a particular trial may be fine for registration, we should be looking at this as in my opinion, a very broad series of opportunities, both clinical and humanitarian and scientific. We now have an enormous population that is already pretreated. Prophylaxis is a misnomer. These are unlike trimethoprim sulfa, where we're giving small doses and it works very well. We're giving out of the starting block therapeutic doses as soon as patients either become neutropenic or become at high risk. So we've lost all that population. That said, there are many institutions, less so in the United States, perhaps more so in Europe, that have elected more for a preemptive approach, wherein we do not have the stumbling block of prophylaxis, and our concept of intervening in acute invasive pulmonary aspergillosis may be optimized. There are individual organizations, Oliver's group and many others that have been very successful in identifying the populations. In India, when I chaired the DRC for the combination therapy, the superb high-quality data that came out of those institutions was admirable. And so I think looking globally with MSG collaborating with other partners in patients not on prophylaxis then allows us to open up some very key questions. Two key questions in the acute invasive pulmonary aspergillosis neutropenic population would be very much, Tom, to your point of endpoints. With isavuconazole, we only have to look at that as well. It took 5 years to enroll approximately 500 patients, 1 to 2 in 103 centers. And the cost is enormous. It doesn't necessarily have to be that way if we can have constructive dialogues with our regulatory colleagues and have informative phase 2 studies that are robustly looking at alternate endpoints. And from the early seminal work of Elianice and all the massive preclinical work we have showing therapeutic response on galactomannan, we should be looking at that as a potential therapeutic endpoint. That would be, and then one could ask key questions of monotherapy versus monotherapy or monotherapy combination therapy. But if I may just add, we have other vulnerable populations. We have, going back to David Stevens' earlier work, we have allergic bronchopulmonary aspergillosis. We've established a small center now for the greater metropolitan area, and we receive multiple calls from all over of wanting help with ABPA. And David, of course, David Denning, if he's here, would advocate for CPA. I'm concerned about CPA for endpoints because this is a chronic disease and you're hoping to stabilize it and we would have to have a totally different set of endpoints. So there are these other populations. I would just add as a caveat, though the ICU population is helpful, when we say ICU, it is enormously diverse, and therein we could have a huge amount of variability.
Let's give the opportunity to some other people to comment also. I would like to ask here our seasoned clinicians here, in your practice, how many of these patients could fit a protocol? How many of these patients are clean cases to be enrolled in a study? Because to me, and we have in our candidemic cases, 85% could not be enrolled in a clinical study. For Aspergillus, even more. But all of this is good and everything. For ICU, everybody agrees on that. But in your practice, how clean of a cases of aspergillosis we have in 2018?
Very few. Mm-hmm. Very, very few. The protocols are really unrealistic. Like you said, 85%, I would think along the same lines. So even when you finally finish these protocols and the data come out, they're really not translatable to the population we are treating. So we're living in two different universes here. We have to make the protocols realistic to where we are at right now.
Yes. A very interesting study design. I was last week in Switzerland that I really like what these people are doing. Everybody realizes that maybe mold active prophylaxis is overused, and everybody's what is quote high risk is not high risk. So what they're doing is they're doing a 3-to-1 randomization. People up front get pentraxin, which is, I think, the molecule that has the most preclinical validation as a risk factor for invasive aspergillosis in allo-BMT, leukemia, chronic aspergillosis syndrome, solid organ transplant. And they go 3-to-1 randomization. If you have a pentraxin polymorphism that puts you high risk, you're put on posaconazole. If you're not, you're put on fluconazole and surveillance. And I think, to me, I'm not saying this is the best study ever that can be done, but those type of studies can also decrease the N and be a little bit more the next generation of studies. I really like what they're doing there. Any comments on Dr. Hassan?
Okay, my point of view is really the practical approach of how you're going to do a study at our days. everybody recognize all our patients on trials. So we have to make the inclusion and exclusion criteria up to date, because right now, it's not, if you want to do invasive aspergillosis, everybody excluding patients on any prophylaxis or any antifungal mold active agent with 96 hours. That's not going to be able to do any enrollment of those patients with that criteria. So this is number one, to loosen up the criteria, because a lot of patients on protocol. In addition, all the patients are on protocol with leukemia protocols or chemotherapy protocol, so they will be excluded because our protocol that you're going to design excluding investigational agent. So those are main point approaches. The other point I would like to mention, really, it's important to see where is the unmet need. So we have now ISA, we have VURI, we have POSA, we have all other agents. So if we're going to pick a trial, we need to pick trial to fit the need that not delivered.
So can you share with us how many patients you enroll at MD Anderson on the POSA versus VORI study? FYI, we have about 60 documented by culture, not galactomannan aspergillosis case at MD Anderson. How many patients you enroll on the VORI and over how many years?
In the last probably, since the POSA VORI started till now, we have high number of patients enrolled, but it's very minimum to the years. So we have 21 patients enrolled in that trial in the US. I am sure there's no other center in the US who are able to enroll that many. But the main problem, what I was saying, is really the inclusion-exclusion criteria.
Okay. So Barbara, you had a comment? Or?
I was just thinking, in the setting of a lot of prophylaxis going on, and the molds that we see breaking through are not just Aspergillus, right? So at Duke, in our experience, when we look back for 5 years at our microbiologically documented mold infections, a third of them had broken through either VORI or POSA prophylaxis, and we did see. And we were seeing more of these azole and multi-drug-resistant pathogens, so Lamentaspora, Scopulariopsis, Pacilomyces lalacinus that we heard about this morning. So I think, you need to take into consideration that these patients are heavily pretreated, so maybe we are seeing a total fewer number of Aspergillus cases. The Aspergillus cases we saw breaking through are actually Aspergillus eustis. We saw that come out as our dominant pathogen instead of Fumigatus. And so somehow taking into account these other historically rarer molds and including those into the protocols now would be an important thing to do. I also definitely agree about having the prior enrollment into a clinical trial with these, with their experimental chemotherapy agents that they are almost all of the patients are on, and having that not being an exclusion is an important issue.
Yeah, a very good example to the new era of aspergillosis risk is we don't know the denominator ibrutinib treated patients in hosts that are considered intermediate or even low risk, like lymphomas. So these people, you cannot use azoles very easily because of tremendous drug-drug interactions. So maybe somebody say, okay, let's assess the risk to the best we can. These patients are not on azoles, and let's design this host-specific group with a new agent. I don't think you need combination therapy for lymphoma patients. Everybody would agree that does have the drug-drug interactions and covers. And in this patient population, aspergillosis has been the main culprit, and pneumocystis, that's the two. So maybe you can pick an agent, for example, that covers well those. I think we need to think creatively the emerging risk group, the drug-drug interaction with azoles. And the other thing we haven't talked in the morning, if I want to play the devil's advocate, we haven't reached the therapeutic ceiling effect of the existing azoles, like what happens if you increase posaconazole dose to 600, 900, so savuconazole? Is there a dose effect to this? That's another population in our hospital that we experiment with dosing. So it's, but I think the ibrutinib-treated patients, if we know the risk, it will be a good study to do, a focus study on this and describe outcomes.
I think that it's very important to understand clinically there are all of these new biologic agents being used. And in the discussion yesterday, I think it was with Candida, how these drugs are impacting the risk for fungal disease has not been thoroughly vetted out. And I think one key issue is for us to come together to figure out how to monitor these groups of patients. And a classic example right now is the issue or the case of ibrutinib and the association with invasive fungal infections. And I think it's important for us not only to describe this association, figure out what the epidemiology is, and then help guide and partner with our oncologic or other colleagues, different specialty colleagues, to figure out how we need to be protecting and perhaps prophylaxing in those patients.
So, if I may.
Yes, Tom, and
just-
then Dr. Tadam.
So, in two comments. One would be pertinent to the small molecule inhibitors and the antibody.
Use the microphone closer to you.
And pertinent to the small molecules in the antineoplastic antibodies, which have immune modulating effect, the Multidisciplinary Association for Supportive Care in Cancer, MASC, these are the same individuals who brought you the MASC criteria under the direction of Jean Klostersky and a team of supportive care experts, already have a full-blown registry for assessing this for invasive fungal infections and several other, as well as bacterial infections. We may be prudent then, and given that they have already made some advance, have the registries, to partner with them. And I think partnering internationally is ever more critical. So I have copies of the protocol. We could easily initiate that. I think a registry for that would be very nice. Building upon Demetrius's elegant paper in CID, this could really help in real-time basis, because these new molecules are coming on a real time. And rather than having just the traditional registry going into remote data entry, we could literally have real-time data accrual and analysis that potentially could be publicly accessible, so that with obviously some editing and proper management, so that the world can see that with this partnership of MASC and potentially EORTC as well as MSG, that we're providing a real-time basis and not of these new interactions. So I think that could be very helpful.
Thank you, Tom.
If I just may suggest one other, we have a wonderful potential for the fifth group of patients for aspergillosis, and it goes back to Barbara's point of breakthrough invasive mold infections, and offer, for example, as an orphan disease concept, not just aspergillosis, but breakthrough mold infections, which almost invariably are going to have high MICs. If it's Aspergillus fusarium, Paecilomyces, Lamentaspora, Scedosporium, that can collectively be said, you have a breakthrough fungal infection, and this is becoming an increasing problem, then you can start. And with regard to the blockage of investigational drugs, usually the oncologist, stem cell transplant physicians will say, okay, you're off this investigational agent until we can get this done.
Okay, thank you. Sanjit, and then Michalis.
Actually, I was just going to say what Dr. Walsh said is about these, breakthrough invasive fungal infections. That's a big issue we are seeing. So for the most part, in the U.S., as we all know, the patients are on prophylaxis, and that's true for our center too. And mostly it is triazole. And what we are seeing on grappling with is resistant aspergillus, things like Lamentaspora and whatnot. And I think that should be a big focus of the trials too.
Yes.
Yeah.
Michali,
I just wanted to follow up on Barbara's comment.
speak louder.
So I think it's important to try to understand the mechanisms underneath that. So what I can just give you as a follow-up, both at the clinical and at the research on ibrutinib, where NIH was at the center of this study. So there is now provision for isavuconazole prophylaxis in the context of the ibrutinib-based therapy for the primary CNS lymphoma patients. The incidence was 40% in that first trial. It included a lot CNS disease. And at the time, there was no mold-active prophylaxis for the reasons that of drug-drug interactions. Now, the reason why this is important to the primary team is that they now have follow-up patients of a lethal disease. You all know that primary CNS lymphoma refractory to therapy has about a 2-3 month survival. They're now having data of those who were treated with this disease that have 3-4 years free of disease completely. So there is an ongoing study following the study, but with the modification of adding isavuconazole. It's still early. We'll have to see what the first couple of patients have completed without getting aspergillosis. At the mechanism-based level, now, and that's where the scary part, I think, we were discussing about this yesterday a little bit too, is when you think of BTK, what we know about BTK, which is the target of ibrutinib, is well known for B cells. And what B cells do to aspergillosis is nothing. We don't really care whether we have or don't have B cells. And many of these molecules and inhibitors have effect on the immune system that are not predictable. And this is a perfect example. So work we've done in the lab the last couple of years in mice and in patients with XLA, which have BTK mutations from birth, who don't typically get aspergillosis, and also in patients who are in that trial and where we can get their myeloid cells before and after they're in ibrutinib, uncover surprising and unexpected effects of these molecules and this pathway on neutrophils, monocytes, that you would never predict based on what we knew from people who looked at B cells and they really didn't look at other places. So I think that's going to be requiring a lot of work. That's just, one molecule and one lab that is studying it. I think we're going to see a lot of those. And we absolutely need to understand, as Barbara implied, not only let's put the drug to cover it, but what's really the mechanism so that then we can risk stratify, prognosticate, do targeted prophylaxis.
Okay, thank you. Any other comments? Let me ask, yeah, go ahead.
Hi. So John Rex over here. Listening to this, a lot of good ideas. Let me suggest something that I think MSG could do that would
Speak closer to the microphone.
could do that would move. Thank you for reminding me. Behold, it is funny how the distance it seems so close. A lot of the ideas that have been discussed are things that you need to filter them through what the current agency perspectives and behaviors. And I've spent a lot of time in the antibacterial world over the past decade, and the lessons from the antibacterial world applied directly to trial design in the antifungal space. There was a workshop recently on narrow spectrum antibacterials which would apply perfectly to rare molds. And there are designs that are eking forward. But you do always have to keep in mind what the agencies are looking for. And at the end of the day, they're looking for an endpoint around how something a human being can feel. And so the mantra, feels, functions, survives, is always the mantra for endpoints. No one ever gets up in the morning and says, oh, my galactomannan levels are so high it really hurts. You just don't say that. You say, I can't breathe, or I'm coughing up blood, or whatever you're doing. And so you have to be really aware of that. Now, it doesn't mean, though, you can't use a different diagnostic, but you have to qualify it carefully. And there are only a very few instances where a non-clinical endpoint is accepted as an endpoint. So HIV is one, and the thing that happened there was we connected HIV in the blood to ultimate outcomes, clear linkage. It's on the fundamental pathway of the disease. I think MSG could, in theory, do that for galactomannan, for Aspergillus, but you're going to have to work at it, and you really have to spend time understanding how you connect one thing to another. And so I just think it's a core function this group could do for the whole industry, and I mean the whole area, the whole academic industry and the industry. It would be to qualify the diagnostics as diagnostics, but also as endpoints. And you're going to have to link them to clinical outcomes. And I think a lot of that data exists, but it's not up to the agency to put it together. It's up to us to put it together in a way that actually works for them. Their job is not to create new pathways. That's something that we have to bring to them.
Very nice.
Demetrius, if I could comment,
Yes.
please. 2 years ago, when I gave the presentation on aspergillosis and future directions, I dedicated approximately a third of it to endpoints in galactomannan, reviewed the literature, and with the blessings of Pete and Peter Donnelly, even created a draft manuscript. But then there was a sense that the time wasn't right I think the time is right. We continue to hear this again, again, and again, and I would ask that we could please, as a group, take the fortitude and the tenacity of pushing ahead, compiling everything that we know about galactomannan and therapeutic response and moving it forward. Similarly, an HIV-infected patient is not going to wake up and say, gee, I feel better today, my CD4s have increased by 25%. The same too is that we can relate unresponsiveness of galactomannan with far more preclinical data than what was in HIV and a very robust clinical data set for outcome in aspergillosis. I'm happy to circulate that draft, and I think we should move it
we know
forward.
the question for you?
Yeah. Randy Walker at Mayo Rochester. I wanted to just say that we also have that quantitative model for CMV in solid organ patients. So it's a powerful tool to begin to get numbers and track response to therapy, both clinically and in assessing new drugs. And the other thing I wanted to touch on was the CIBMTR organization who follows our allo stem cell patients. This would be a group who have the high risks, have subgroups, of course, who need mold prophylaxis frequently. And this group is very closely monitored, and they have to report their outcomes. So you have the ability to look at cumulative incidence of infections in this group. There's a lot of people capturing clinical data in real time, and I think that would be another organization to partner with. And then on the topic of real time, with multi-center collaboration, you know, we all might see 10 or 20 or so cases of Mucor, for example, per institution per year, but with a multi-center collaboration, we could compare year by year by year with cumulative incidence. Are we moving the needle or not? You could use big data analysis to compare different centers and say, oh, these groups over here are using POSA, these groups are still using surveillance, et cetera. So I would follow up on Dr. Walsh's suggestion for collaborating with the hematology groups, including the CIBMTR, and also thinking more about real time. Thank you.
Yes?
I just would like to suggest that the opportunity is
Closer.
The opportunity is even larger than just galactomannan. So medical mycology is interesting in the sense that it has an abundant number of biomarkers that can be used to follow therapy. So galactomannan's one, glucan's another, quantitative counts in Cryptococcus is another, and quantitative counts in Penicillium is a fourth. And the double-edged sword is that, unlike bacterial disease, you never get the organism. So the opportunity, I think, is to radically redefine or use biomarkers, not only to assess outcomes, but you can use that to assess, integrate the MIC, which you don't get from drug resistance, Tom. You can get a read, and what I'm trying to say is you can get an in vivo MIC, actually. It can tell you about how the host and the organism are interacting, and the tools are available to do that. We've done this with galactomannan and doing it with quantitative counts, done it with CRP. So I think that the whole field could be radically changed by using biomarkers.
Dr. Messere?
Yeah, the problem is that with all the biomarkers that we are mentioning and with the HIV and the CMV, you see that it's more clear-cut. In my mind, it's the context where these tests are being done is cleaner than when we are trying to apply the galactomannan in these patients. When it's not a perfect test, and there's so many co-founders to the results what we see, the way is it used, and I think the issue of a massive use of prophylaxis also is a big problem, which you don't have probably in HIV or in CMV or when you are trying to diagnose crypto, monitor crypto, because you really know what you are going after. In the world of galactomannan and even PCR for Aspergillus or beta-ardiglucan, that's a problem too.
Yes, so I actually will disagree with that. For a single point estimate of galactomannan, the variant structure really sucks, right? And anyone who's done that knows that. But if you have 6 samples from a patient, you have a very good idea about how they're traveling with antifungal therapy, and if the trajectory is going down, they do well. But you know, if they're going up, then you have to change. And so this is a way not only to individualize or personalize therapy, but it's also a way to develop new drugs.
Mm-hmm.
So let me challenge you a little bit on the galactomannan, and I'll try to be the devil's advocate here. What do we know about galactomannan producing pneumonia that looks fungal? What is the bug in the era of we don't do biopsies, we don't do autopsies? We published our results about breakthrough infection to asafuconazole, and 50%, and the typical breakthrough infections were people with 0 neutrophils for months, active leukemia, who died with a terminal pneumonia with multiple masses or nodule lesions with negative BAL and negative galactomannan, and the very few people we had cultures were mucorales. So what I'm trying to say is, if you are going to design a future study with mold-active breakthrough infection with a galactomannan-producing organism, and reconcile what Barbara said, that most of these are weird things, cryptic Aspergillus, we don't know the kinetics. Most of the kinetics of galactomannan have been studied in Aspergillus fumigatus, to my understanding, or Fusarium that produces galactomannan, Galactomannan scedosporium species. Is galactomannan positive, CT positive, host positive, synonymous on Aspergillosis study? Can we burn a good drug for Aspergillus, but design a study that is focusing on galactomannan as the biomarker for Aspergillosis?
I think we're looking at an apples-oranges situation. So if you have that, we talked about 5 major populations. The breakthrough population is going to be, by definition, heterogeneous. And given prior antifungal prophylaxis, it's not clear whether the Aspergillus breaking through at that point is going to be galactomannan. I think one should take that off the table. One would want to use, in a much more pristine population, the classic AML population that is not receiving antifungal prophylaxis, of which many institutions are not. They would use typically a preemptive empirical approach with a fluconazole base. And in that population, if typically one does see rise in galactomannan, and as William just said, when you look at the data for 6 or 7 consecutive points, you see them coming down. Where that was especially compelling was in the combination study. When we reviewed those data in DRC, voriconazole, inidulafungin versus voriconazole, multiple patients, because they had stable disease by radiographic, but although resolved their clinical manifestations, were called stable equals failure. Yeah. But if I may, what came down and correlated very nicely, and a number of you were on that panel, was the galactomannan. It came down very nicely, but we could not use it.
Yeah, I think you missed my point about early on about that study that might not be pertinent today, but my IRB is going to tell you if we're going to go, how come these guys not on posaconazole, it has been shown to be associated with improvement. How I'm going to tell my IRB about what you're saying? I mean, there is a slide I saw in the morning that at least in the United States, most of the tertiary care cancer centers have prescribed to posaconazole prophylaxis, endorsed by guidelines. Randomized studies have shown survival advantage, and that's the new name of the game. And in this population, actually can be relatively homogeneous, not heterogeneous, what breakthrough infection is.
So, Demetrius, in that regard, you're absolutely right. In the United States, largely posaconazole prophylaxis is used, but I could list the names, including a number of colleagues who were on the New England Journal paper, on the pivotal study, who do not use prophylaxis. Yes. It's an institutional decision. And so respecting those institutional differences, one would then want to say, if you did a survey in terms of qualifications for, say, study 1, study 2, if you're using broad-scale prophylaxis, you're not going to be enrolling patients on the acute study. You enroll your patients on the breakthrough protocol. and then in which galactomannan probably would not have. Maybe next generation sequencing, liquid biopsy, cell-free might be possible, but that's very far down the road. But then you would then have the other institutions, many of whom would not be in the United States, with whom we could at least partner, India, Europe, others, where they made institutional decisions, and we will take a preemptive approach. And if you look at John Wingard's study of voriconazole prophylaxis that was versus fluconazole, you may say, well, it's obvious that you'll have more breakthrough on fluconazole. No, everybody was backed up for equipoise intentionally with empirical therapy with liposomal amphotericin B. And what came out of that was that equivalent outcomes, absolutely, virtually equivalent. So in that regard, one doesn't necessarily have to be completely committed to POSA. I'm not trying to in any way suggest changing institutional strategies, but there are many institutions who elect not to use POSA prophylaxis.
It's a complicated issue. In a Wigan study, most of the patients was lower or medium risk of death. In our institution, most of the people who went to the study was a bunch of myeloma patients. When we present the study to all BMT colleagues, they say there's no way I'm going to randomize to fluconazole and hope for the best with galactomannan. What you're suggesting, Tom, and I'm not disagreeing, is a study that is going to go to Europe with by a market-driven approach, or India or Far East. Since we're in the mycosis study group, my concern is how you're going to design a U.S. meaningful study when at least 70% of the patients, number one, are on mold-active prophylaxis, and second, there are very few people who can be eligible have comorbidities that are not able to be put. So there are very few people, and they're very selected people. So that's my point.
So in answer to the question, it's absolutely, if we're looking at broad partnerships, then we need to be thinking strategically, if even an experimental clinical trial design with our partners in Europe, India, elsewhere. That does not necessarily mean we take necessarily a lead, but at least we have an intellectual contribution. If we look at the situation here in the United States, where most of the institutions and most of the high-risk patients are being treated with mold-active prophylaxis, we then need to say that the challenge here is one of resistant molds, and be able to say it's, for example, the cryptic Aspergillus species, Fusarium, Lamentaspora, Scedosporium, Scopulariopsis. And that's as Ray said, that is the unmet need right now. These are breaking through, and everybody understandably shrugs their shoulders, and we go to liposomal amphotericin B, but that's all we have at this point. So there is a rife opportunity for these breakthrough invasive fungal infections, which is really dominating the picture. Or we go to these other groups that I mentioned before.
So, Demetrios?
Yes?
So,
Go ahead.
you can take a page right out of the antibacterial universe right now. Most of the big antibacterial trials are about 5 to 10% US, and the rest is ex-US because of a problem like you're describing right here. Mm-hmm. That's still okay for getting drugs approved. The FDA, understands that challenge and knows that if they were to insist on it being a 30% U.S. trial, nothing would ever get approved. Mm-hmm. Perhaps something that this group could benefit from would be to bring a regulator, and they're very, Somani Nambiar and Ed Cox are very willing to turn up at sessions and have conversations, and talk about the nuts and bolts of trial design from the agency perspective, so that you've got your heads wrapped around what they're thinking about. The times when we've moved the needle in the past few years, it's when we've learned to look through other people's eyes, and particularly the agency's concerns about how, they have to think about precedent, they have to think about knock-on effects in other settings. But a trial for the U.S., no, nobody's going to do a trial just for the U.S., but you can get enough experience, and I don't think that you have to have a large number of, the United States is a giant mixing pot anyway of backgrounds in terms of pharmacogenomics. I don't think it's that big of a deal that this is only 10% U.S.
Yes, maybe an idea is to look a little bit more carefully the way very expensive oncology drugs are approved, especially checkpoint inhibitors, and if you see they have accelerated approval by FDA, and if you see carefully, their endpoints are kind of soft. They are not based on survival advantage, but time to relapse, time to progression, time to this. And we never thought a little bit about if we say aspergillosis is a complex subacute disease like cancer, time to relapse, time to progression, instead of looking for total outcome that is a little bit unrealistic, I think, in 2018, because we have very comorbidities, competing cause of death, the driver is underlying disease. So maybe we should think how the oncologists think, and then get the drugs approved. Any comments?
Yeah.
Yeah.
Me.
Just a comment in regards to in the new era or really breakthrough infections while on prophylaxis, on mold-active prophylaxis. So it will be quite interesting to hear from you. What type of approaches you would like to have tested? So do you want to see how these patients will perform if you switch them in a randomized way to amphotericin B versus a totally new experimental agent? Would you like to see the approach that how about adding on something in the meantime that you figured out that this is a true breakthrough infection, or what were the levels? What are the type of approaches that you would like to really see being explored as a therapeutic approach in these patients that are breakthrough infections? So it's add-on therapy, switching entirely to amphotericin B versus switching on a new investigational agent. What's the type of designs that you kind of could suggest so that we from the industry can think about if we can fit into those type of paradigms, treatment paradigms that you're envisioning that should be explored?
Yeah, absolutely. That's what I was thinking, actually, that we are trying to fit studies, in my mind, that to do in the U.S. And as we said before, the need seems like the need we are managing a different type of problem here because our reality is different. So we are having problems trying to accommodate trials that really don't fit us. And we all have experience trying to see, can we do this study in our institutions or not? And for this or for that, we cannot do them, right? So probably we are dealing with a different reality, and we need to see what that reality is. For example, the breakthrough infections, we don't see too much of Aspergillus resistant to azoles, but we see other molds that are resistant to the azoles we're using. We're seeing a lot of Candida species that are resistant to mecafungin or azoles that we are using. So these are the breakthrough type of infections that we are seeing. And probably we can provide the answers to those questions that through studies, right? Different type of studies that the industry probably is not making those studies for us. So this is what I think is the discussion needs to be healthy in that way that, for example, we said that there is no resistance to azoles in many of the US, but do we now? Do we really know that? Because when we order, we send it to these species to Texas to see if there are, to see the MICs, we are actually it's a catch-22 to me, because we really don't know what's happening in our institutions in that regard. Is it the time to do screening for the resistance, do a quick plate test for resistance at front, and then decide who do we send to San Antonio or not? There is a lot of non-answered questions in that regard too, and that's an unmet need, I think.
There is if I may, when we speak about resistance in breakthroughs, quite often we'll measure MICs and we'll not see significant elevation of the MICs. So there may be, quote, resistance as defined by perhaps an epidemiologic cutoff value or interpretive breakpoint, but there's this very critical factor that Tom had brought out of host response. And profoundly immunocompromised hosts can have a breakthrough of a fusarium, and you look at the MIC, and maybe the MICs aren't sufficiently predictive, but it's not like they're necessarily off scale. The mucorales will march right through. We've seen it happen on isavuconazole. We've seen it happen on posaconazole, yet the MICs are still not off scale. So many of the interpretive breakpoints that we have do not take into account the vast, profound degree of immune suppression. But the problem is, and it is the unmet need, we do not have a treatment paradigm. We do not have a uniform modality that we can apply to the breakthrough molds and their devastating complications.
So Nathan, what do you think?
No, I would agree with that comment. You know, in the fungal world, we have very few breakpoints established. We are now trying to move toward ECVs. But you bring up a good point. Do we need to have patient group specific breakpoints in the future? So we all know that a low MIC is not a predictor of success, especially if the patient has no white blood cell count or is on high doses of corticosteroids. So these factors need to be taken into account. I just don't think we have the data accumulated yet to go forward with it. With molds, we don't have any breakpoints, regardless of the patient population, regardless of the drug in the United States, and very few are set in Europe. So if we are going to move forward with that, there's going to have to be a lot more data collected and analyzed in order to make it meaningful.
Barbara?
So putting on my micro lab hat for just a minute, I think what you said is true, right? So the antifungal subcommittee of the CLSI have been working diligently to try and for the opportunities where it's possible, and I think the one combination where we may be getting clinical breakpoints soon is voriconazole and Aspergillus fumigatus specifically. Whether or not we have enough data for isavuconazole, I'm not sure. But then for the other organisms where we don't have clinical breakpoints and probably won't have because of lack of clinical outcome information, I think that's where the ECVs become critical. And so obviously, you know, Nathan, and many others in this room, Sean, we're all working very diligently to try and put ECV data together. And so how can the MSG help? It's help give us these isolates and the MIC data on those isolates so that we can help establish these ECV values. While the ECV values will not tell you that your patient's going to respond, they don't predict response, they at least help us determine when the isolate is behaving differently and is beyond what is typically represented in the wild type population, and so therefore might harbor a resistance mutation. And I think from a practical standpoint on the ground, that's a very much needed clinical tool, right? So I think as the MSG helping the CLSI antifungal subcommittee move the ECVs forward, then, you know, as we gather that data for the mucorales, for fusarium, for these other important molds, that's going to help us, right? To get to your question, David, I don't know, but when we're thinking about the breakthroughs, there's many different reasons for breakthrough, right? There's subtherapeutic drug levels, there is a host without any immunity, without any cells, and then there are resistant pathogens. And somewhere it seems like we should be able to, if a person is on prophylaxis and they're breaking through, somehow characterize what those various groups are, randomize them, perhaps to liposomal amphotericin B versus a new agent that has potential activity against these pathogens, and then we can, you know, I don't know how to define the group, but it seems like we could put our heads together and put up some kind of paradigm where we're taking these factors into account to be able to put these patients on a clinical trial.
Also, with respect to
one step. I'm sorry. Please go ahead.
Just for the ISA, there is a manuscript which David Endes is the first author, which addressing breakpoints in relation to MIC, which should come out soon, hopefully. You know.
The other thing, wait, let me just say one other thing. The other thing that I think for clinical microlabs, so susceptibility testing is usually performed in select laboratories, but I think another key issue is making sure we know what the pathogen is, right? And so we've heard repeatedly where the pathogen has been misidentified, and the molecular identification of these organisms, what we thought was Pacillomyces isn't even Pacillomyces anymore, et cetera. And so the target is moving, or phylogenetic classifications are unstable. But I think one technology that is increasingly used in clinical laboratories across the country for bacterial, candida, and crypto identification is MALDI. And there is a lot of work going into the use of MALDI for identification of molds. I don't know that we're there yet. I think the Vitec technology, their MALDI instrument may be a little bit ahead of the Bruker, but I think that is something that we can all actively work on right now to increase these reference databases bases in these MALDI instruments to get us to at least the point where we know what pathogen we're dealing with when, in fact, we have an isolate.
With regard to the concept of liposomal amphotericin B, we could potentially look at this as recognizing the critical need for new developments in breakthrough fungal infections. If we could take a standard protocol that would address this and have basically a platform where the gold standard would be, it's debatable, but I think most of us would agree, would be liposomal amphotericin B through breakthrough on mold, anti-mold prophylaxis. One could then evaluate several drugs, either consecutively, simultaneously, and be able to accelerate using the gold standard, liposomal amphotericin B, in separate individual clinical trials, non-competing, but really usher in a whole new approach toward these with these new agents. If you look at the Tango 2, for example, in meropenem vaporbactam, they targeted carbapenem-resistant Enterobacteriaceae very specifically. Although there are limitations to the comparison, you could then also say that we could target in appropriate controlled studies using liposomal amphotericin B versus the study drug. And that could give us an iterative platform that we could use to study several agents.
So, Tom, let me challenge you a little bit about a uniform prophylaxis. Maybe I haven't been smart enough to figure out. Let me give you an example of a real patient. You're called on a Thursday afternoon or Friday afternoon about a patient with 5 nodular lesions. Bronchoscopy might not be done on Friday afternoon or might be postponed on Monday. On Friday afternoon, the patient is moderately sick and you need to make a decision to start something in the preemptive space. In the best possible scenario, the patient has bronchoscopy on Monday. In our institution, we have same-day galactomannan results for BAL. So you know he has a galactomannan-producing organism. We already said can be different things in the breakthrough space. Culture, multi-TOF, we have multi-TOF also based on node might be available a couple of days later. And resistance, if we're going to send the isol, is going to be in about a week after you make a decision. How you're going to design a therapy and evaluate a drug when you're going to make a decision on a Friday afternoon and start ambisome, that's what we usually do, about a new drug that you're going to enroll the patient on next week when this critical thing is there. How you're going to do that? So
we encounter that commonly, and certainly the clinical trial gap, which can be as much as 3/7, as much as 42%, the Friday, Saturday, Sunday. First of all, you have to have a dedicated clinical research team, people that are going to be staying there on a Friday. Apart from anything, personnel is absolutely essential. And when we look at low enrollment, it's often because institutions may not have that kind of infrastructure. That would be number one. Who's going to stay on the Friday afternoon? Number two is that we already have a footprint for this. We already have a template. If you look back to Oliver's study, Ambilode 3 versus 10, this was, to my knowledge, the first study that really did this, and then it was subsequently emulated by the Vori Inidula fungin study, where basically you enroll patients initially on the basis of a possible infection, possible breakthrough. All they have is host and nodules, but you enroll them on that basis to be followed with, and randomize them to be followed later by subsequent definitive studies, be it percutaneous needle aspirate, be it VATS, be it PCR from the or identification and culture. But one can enroll those patients, capture them, and if there is negative data completely, then you either convert them to standard of care or you can still follow them on study, but not in the randomized per protocol analysis. So we have that template. It worked elegantly for Aspergillus in 2 major studies and several others, smaller ones of which I'm aware. And why not apply that early approach to be later confirmed for the breakthrough infections?
I understand that totally, but again, let me make it a little more specific. David asked the question, I want to use Abraxa in this patient preemptively. And you have a patient who has a breakthrough infection, especially in some places that you don't have the rapid diagnostics to know what the fungus is. When Abraxa can be a great drug if it is going to be a nasal-resistant Aspergillus or other things, but Abraxa, we know what you said, about 40% of patient studies can be upgraded from possible to probable based on subsequent workup. But sort of having the pan-mold agent, and some of these drugs here presented might fit the bill, might not, but you may have a beautiful drug for a specific bug, but cannot be used safely in the preemptive space because the differential diagnosis of mold, multi-drug resistant mold breakthrough on a very good drug as a posaconazole can be very broad. I think that was his question, and I think most of the people go to liposomal lymphoteresin B as one pathway that I think will be one arm in the preemptive decisions. And in my opinion, we should have a very rapid diagnostic to know what the fungus is, and then maybe then feed off to different drugs depending on what the fungus is. Otherwise, it's going to be a very heterogeneous.
Well, we have to look at each individual agent. Not all of the candidate agents that are now available are going to have sufficiently broad spectrum to be used empirically for breakthrough fungal infections. In that regard, either we had a rapid diagnostic to say, at the bedside, to say yes or no, it's not. I'm concerned that we do not have the spectrum of analytical capability at the bedside that would allow us to be able to distinguish a breakthrough Aspergillus from a lamentaspora.
So do we have that?
But
No.
You could not, I don't think we have that at the bedside, period. So given that quandary, I think we are already saying that by design, if you're going to be empirically or preemptively initiating treatment for patients that are going to have breakthrough mold infection, suspected breakthrough mold infection, you need, by definition, a broad agent. And that then would bring you back to say, okay, if we have a more narrow spectrum agent, then we're going to then have to put that agent or study that agent in the patients where we have strong data, be it galactomannan or others, for invasive aspergillosis, using it in a targeted Aspergillus naive population.
Yes?
So discussion the issue of the agent that need for the unmet need. Certainly, you know, everybody's been talking about breakthrough. That's one point. The other point, we have a problem also, need to look into that point for the new ideal agent, drug-drug interactions. So we have a lot of patients, drug-drug interaction, especially now with new chemotherapy use, a lot of azole cannot be used. So really appealing agent is going to be good agent, doesn't have drug-drug interactions, very sidal agent, and then can be used on those settings in addition to breakthrough fungal infection. One other point is really dramatic, what Dimitri was mentioning. When can I put that patient on a new agent? All right now, either any FDA regulation, they want you to use probable or proven fungal infections. There's no way you will be able to have that decision within 96 hours. And that's why the major problem, you lose a lot of patients. So if any way we can release this to at least 1 week, you will be able to really include more patients in that setting.
For entry, we are allowed, and several studies have now done this, and with regulatory approval, we are allowed to enroll patients with early disease, meaning possible, as long as one has built in, usually within 72 hours, a definitive assessment, be it bronchoalveolar lavage, biopsy, culture, so forth. So we do have that entry, and that has been, I think, a helpful tool. What does serve as the real problem, though, is this continued, and I'm not sure what the regulatory pathway could be, would be the restrictions, since many of our patients are on protocol, that if they're on an investigational agent, can we bring in another investigational agent? And that becomes a great dilemma.
Any other questions from the audience? It's obvious it's a very complicated area for drug study design?
Looking at it from a completely different viewpoint, these are relatively unusual diseases, rare diseases. We are struggling to enroll them in patients, enroll them in studies. And MSG is confined to the United States. I think we have to look broadly across the world. There are a lot of mycoses around the world, and MSG should take the leadership and assure the pharmaceutical companies we will find good centers in Latin America or Africa or Asia, reliable centers to do these studies. I think these companies would be happy because we would be the intermediaries in finding reliable centers to do these studies. I think we should break our shackles and think broadly. That way we can do these studies successfully under the leadership of MSG, and the quality would be still very high. I think we need to think along those lines.
Chandra, building upon your suggestion previously of the Indian Society for Clinical Infectious Diseases, could you imagine if we opened up free membership, not the $100, but free membership to that entire group and say we want, those of whom, of course, have a interest in medical mycology. Just using that as an example, we could identify other groups as well. And just imagine 2 years from now, here in Big Sky, that with electronics, they could be participating in the same meeting, embracing that and extending through the electronic system a global network of clinical trial investigators. So we don't have to anguish to say, well, we need to develop a US-based MSG study. We should be looking to say an internationally based MSG study, and using, from my experience and your experience with the excellent results that have come out from that society, we could certainly start that as our open arm extension for free membership and participation.
Absolutely right. Yeah, and I think we can have an evening or afternoon with the IDSA week with MSG, and many of these foreign participants come to attend ID week, and then they will be given an opportunity to come to the MSG session, which can be an extra session annexed with the ID week.
Since I think we have time, two other issues that we haven't talked in the morning that can be an opportunity or a challenge to use new studies is the developments in innovative diagnostics beyond biomarkers, which is immuno-PET-CT, theragnostics, and immunotherapy. There have been active groups about CAR T-cell therapy expressing Dectin-1, and we have a second-generation agents we're trying now. So it is possible you can address one of the elephants in the room, which is host immunity, and the other is to do a theragnostic study that when a drug can look much better beyond the culture-based, multi-TOF, galactomana. And this is not mature, in my opinion, to be implemented in 3 to 5 years, but maybe it's going to be even more easy or more complicated depending on the point of view. If, say, there is in Europe a macro-PET study when a monoclonal antibody targeting Aspergillus hyphae is implemented for early diagnosis, it will be so cool to have immuno-PET-based diagnostics and starting a drug, say, drug X that is a very good Aspergillus drug, but not very good mucorales drug for this situation and moving the needle in a meaningful way. So there are other things that are coming, and I think MSG should have the receptors upregulated to look at what is going on, plus immunogenetic stratification and everything. So it's not only the drugs. Backing up, I think Tom Patterson was a senior author in a paper we published recently about asavuconazole versus voriconazole, the ad hoc analysis in neutropenic patients with aspergillosis, and we look in outcomes on day 42 if the people did not recover the neutrophils. Both drugs did exceedingly well when the neutrophil counts recovered, aspergillosis, not fusariosis. And both drugs were very mediocre, no more than 25%, if patients were in the setting of profound neutrophil recovery. So I think a big part of the agenda, and we don't know how to do that yet, is how to stratify for underlying disease and neutrophil recovery. And to make things more complicated, in our leukemia service, most of the patients who get aspergillosis in leukemia service are technically non-neutropenic. What they have is 30,000 white cell count with 35% blast. Neutrophil count technically is more than 1,000. These people, if you're going to stratify them based on clinical studies, and I think my co-study group should benefit from having seasoned hematologists, if you're going to do hematology study design that can give you the landscape of leukemia therapy, the landscape of transplant therapy, how you stratify patients of karyotyping of leukemia. I just think somebody in the morning told me he liked my coin term, myopic term. I think we're a little bit myopic the way we look in fungal trials, especially in leukemia population. The other thing we need to talk about is what Chandra said about chronic aspergillosis syndrome in say, India, post-sarcoidosis, post-tuberculosis. This, I think, a very cleaner population to study compared to hematology, and I think we should make more effort beyond hematology to chronic aspergillosis syndromes.
With regard to the chronic aspergillosis population, David Denning and I have had a number of discussions. We've compared patients, and David has given a tremendous amount of thought to these endpoints. And if you just take, whether it's aspergilloma or whether you're taking into account chronic necrotizing aspergillosis, chronic cavitary, the response criteria are going to be extremely different. An antigen is not going to help. Usually they're antigen negative. If you look radiologically, you are happy with stabilization. The discussion is even now down to the potential of millimeters changes in the pleural surface, which is probably not going to rise to a level for regulatory robustness. So I think that is a population that suffers terribly and worldwide from invasive aspergillosis that would really merit an in-depth analysis of what has been historically the response and what we could anticipate in the natural history on voriconazole. There are multiple patients now that have been treated, and trying to hone in on what would be the best endpoints would be the next step in the right direction. With aspergilloma, it's a little more complicated because you have about a 20% spontaneous remission rate as well. But these are challenges in that disease.
Well, I would agree with that. The chronic cavitary and conditions are going to be really difficult to study, and the outcomes are frankly going to be really bad, regardless of your antifungal therapy. The flip side of that, though, there's a huge population with ABPA. I think like after more recent drug development, but also the guidelines, I think all of us, even the ID folks, like we see a lot of people with ABPA. And that's a disease where the markers are very clear about success. And so there you have easy to measure markers, and so you could have fixed points in times to follow, you know, the IgE levels and that sort of thing. And it clearly correlates with symptoms and disease and the use of steroids and the whole 9 yards. And so I think that is a very attractive group to study.
I would agree, and we see patients who either do not tolerate voriconazole, itraconazole, yes, no, they may not respond to, often they go on Vori, and then they are intolerant or refractory to that as well. So this is clearly a disease where the alternative is, and we see some of these patients presenting, frankly, Cushingoid, and with serious complications from long-term prednisone, sometimes on it for 6 months to a year. So there's a crying need for this, both on scientific and humanitarian grounds.
I'd bring up one other little thing about the diagnosis to try to enter patients into breakthrough trials. It seems a little out there, but I'll give an example from our institution. Has been interested in resistant bacteria, and they have been interested in taking prostate biopsies and finding resistant bacteria, and then entering them on different prophylaxis agents, broader agents or narrower agents, depending what the bacteria was, but they need to do it right away. What they were able to do, or what we were able to do, was actually take those samples, the prostate biopsies, and do molecular testing, PCR test, to basically look at specific genes that they were targeting, and actually in the same day, you know, a few hours result, basically enter those patients into a trial with one agent or the other. So I would say that there are a number of centers. Lewis has a lot of centers lined up who have on-site active PCR machineries, mechanisms that I think, honestly, they could do that with biopsies in a fairly real-time basis.
I think certainly in respect to the biopsies, you're sort of in a situation there that you're reliant on the analytical specificity of the PCRs. And so providing they are specific, the test in itself is quite straightforward and can be done quite rapidly as well. I just want to make a point as well. I think one thing that the MSG should be sort of maybe a surveillance study into sort of post-influenza aspergillosis. Pete mentioned he's already had 2 cases this year. It seems quite topical. You've got the network in place. You're also in a situation where you've got rapid diagnosis for not just influenza, but for other respiratory viruses. But you could even take that further. We've noted in Cardiff, we've had 4 very unusual cases of aspergillosis in the summer months, ranging from Crohn's disease through to alcoholics and to even an IV drug user who thought he could fly and had traumatic lung damage afterwards, but they all ended up with invasive aspergillosis. And I think this is some sort of thing we need to know the incidence of these diseases.
We have a question.
Yes. I'd like to revisit our discussion about endpoints for clinical trials. Tom, your comment about looking at endpoints in a study of chronic aspergillosis, including stable responses as successes, sort of reminded me of all the issues we had around the global response in our previous clinical trials. And I think this is an area where we need to think about how we can do that better. You remember that the global responses in the combination study did not match the survival data. So the question is, what do they even mean, given the way that we assess them now? So a couple of issues, particularly when we're looking at early responses, should we consider a stable response to be a success rather than a failure? Also, can we take the last observation and pull it forward? As you mentioned, a lot of the patients in the combination study were considered to be failures in the global response, not because they failed, but because they were missing data such as CT scans within a very tight window. So I'd just like to know what you think about reassessing definitions of global response in aspergillosis studies.
Well, I think it's a good point, but I think it's also helpful if we're able to build in multiple endpoints, so you're not just stuck with one global response, that you're able to look at multiple endpoints, we talk about multiple biomarkers. I'd have to say that biomarkers have just got to be used for these trials. I think that's how we're going to be able to get the endpoints that we need to get the approvals for these drugs.
From a practical standpoint, and we had patients that had not only been discharged, but were graduating from college on the study, and literally they had stable pulmonary infiltrates. Galactomannan had come down, but because the infiltrates had not regressed by 50%, then they were deemed stable. And therefore, I think honing in on that and maybe pulling out that data, if possible, and presenting it as a case at least for aspergillosis in that population might compel a rethinking. And then statistically, it then could maybe shift us away from that, that middle zone where you're enrolling massive numbers of patients just to try to get to a non-inferior endpoint, because certainly it doesn't reflect reality. Most of our patients will go on, even with pulmonary infiltrates. Eventually they'll resolve. But clearly that's one area that needs resolution. Endpoints need further clarification. And within that database, you can see very nicely excellent responses. So I think it would redound considerably to our being able to understand endpoints, and perhaps even in the same process, redefining stable as being successful.
Well, I'd add to that, though. I think if we think back, how do we come up with stable was a failure? And I think how we came up to it, if you think way back in the day, like the caspofungin initial studies looking at salvage therapy, those patients were enrolled often with long-term stable disease. And so it made it pretty obvious that stable in that setting of a new drug added, you really couldn't help but call that a failure. You couldn't call it a success because they were already at that state.
So it goes back to, and that's true. And from my understanding, from a broader regulatory standpoint, is that if you start with symptoms XYZ, symptoms and sign XYZ, you want to see symptoms and sign XYZ resolve to a certain level. And therefore, if the pulmonary infiltrates have not resolved to a certain degree, then unfortunately would be deemed stable. Yet it becomes eminently clear in the DRCs for aspergillosis, what drives the decisions is the pulmonary infiltrates. And I'm not sure that CT scans of pulmonary infiltrates in that 6-week period are sufficiently predictable when everything else is resolving. My suggestion in that regard would be to have a more algorithmic approach. If your CT scan remains stable, but if symptoms all resolve, symptoms and sign resolved, and galactomania resolve, why not call it success?
I think it's also a little bit more complicated. I was a part of DRG of this trial year, and one of the difficulties when you evaluate infiltrates is you need to know what happened to the neutrophil recovery when you evaluate the infiltrate. So if the patient was neutropenic and the data collection form was heavily biased, what I kept saying, to mycology endpoints without information about underlying disease. So in other words, if the patient had stable infiltrates and was doing well and he was in remission versus having stable infiltrates and he was still neutropenic, it's a total different ball games here. Mm-hmm. So I think you need to have information about the underlying disease, neutrophil recovery, reliability of galactomania and kinetics, and when that is meaningful. I don't think every time randomly, maybe we should have a structured approach of kinetics or galactomania, and trying to go around the problem of missing data, because everybody agrees that was not a good way to evaluate a study. But we didn't know what to do with infiltrates. Different radiologists were reading different CAT scans. There is a study in Gestion by Erasmus et al. When they put people with lung cancer, which is better quantifiable as a mass, and they gave it to 10 radiologists. And the discontinuous rate calling radiology response was 40%. So there is a lot of operator-to-operator variability. How this is called, you don't know how the neutrophil count does and how you interpret in this context. I can even be a little bit more bold and say maybe PET-CT is a better way to evaluate, because then you can see FTG avidity and quantify versus infiltrates that can be post-inflammatory scarring in that area. So it's a very difficult question, and maybe we can come to an algorithm, but it remains to be validated somehow. Maybe we can come with an algorithm, but at minimum, I would say, consistency of how you read the CT, consistency of the technology of CTs, extra effort how to evaluate missing data or to avoid missing data, and factor in neutrophil recovery and underlying disease when you make the follow-up assessment.
So Demetrios, you make a compelling case, I think even more so, for our, as a community, evaluating global response criteria. Historically, the mycology community backed into it. We were handed, after the International Congress on Harmonization, it was basically said, here's how you're going to evaluate compounds. And mycology backed into it, and that may work well for candidemia, where basically you have a microbiologic response, there's no very minimal radiology, but when we're looking at mold infections, it needs to be further refined algorithmically, including all the parameters on radiology, host response, in ways that we as MSG could really revolutionize and help to improve clinical trial design and not recapitulate the problems of the past.
Excellent.
Yeah, I just want to reiterate, I think it would be great if the MSG could come up with some kind of position statement on how to assess a global response in a clinical trial and have it available so when we do design these clinical trials, we can at least point to that reference to support what we incorporate in the clinical trial.
I think there is a statement out there, the Bramsiggles et
Yeah.
al. CID, that I think is a little bit outdated, and
Exactly.
I think we need to revisit that.
That's it.
Along as we said before, variability of definition of what is a breakthrough infection. This also needs to be given some kind of crisp definitions because everybody talks different things about what is a breakthrough infection. So I think MSG can come to with this type of help for the industry.
Okay, thanks.
So, any other questions from the audience? Because I think the time, yeah, Barbara?
Time is up. I'm
No,
changing
we have
gears.
time.
Do we? All right. So I just wanted to bring one other issue up that has not been discussed yet. So changing gears just a little bit. It's prophylaxis in lung transplant population. So this is a problem. Standard of care right now is to administer prophylaxis, typically until the anastomotic sites have healed. So that usually is around 3 months, 100 days. But no drug has FDA approval. And so when you're trying to bring a new drug to, well, what that translates is in that the patients often can't afford the medications because it's not, if you're looking at voriconazole or itraconazole or even aerosolized amphotericin B formulations, the third parties don't reimburse. They're not FDA cleared, and so patients can't afford it. So it's a quagmire, right? And then when you talk to companies about, well, how would I do a study of prophylaxis in the lung transplant population? No lung transplanter is going to let you enroll into a study where you don't have them on prophylaxis, so we have to have a comparator. And so what I think is, if we as a group could put together a white paper arguing that antifungal prophylaxis is standard of care, so then that would allow some of these companies to then be able to work with the FDA to perhaps study a new drug, get a drug on the market that is then cleared and perhaps covered by third parties.
Well, I'll add to that. We, of course, did have the survey that was supported by Maine Pharmaceuticals. It's not quite done yet for surveying sort of the landscape of transplanters for prophylaxis in lung transplants, but also in hematologic malignancies. Those are in progress. Hopefully, we'll have results of that soon, and I think that could be a nice sort of backbone for progress moving forward, whether studies could be done. That's part of the survey. Would you be interested in surveys, in a new study? And so hopefully that'll help move that forward, and I think a white paper would be a nice endpoint for that.
So Neil?
Yeah, I can't strongly endorse what Barb said enough. I mean, the issue of patients not being able to afford their drugs post-lung transplant is absolutely huge. Secondly, I'll say there's a ton of fungal disease and colonization in this population, and it's poorly studied. And then finally, clinical practice is a mess, and there's no, and it's not a bunch of people like you sitting up at the panels in the lung transplant world. It's transplanters and really heterogeneous, intellectually inconsistent practice, and it's something that MSG has had less involvement with in the solid organ space, but I think it's really, really, really important.
Pete?
Yep. Every year since we've had this discussion in the Aspergillus space, we bring up lung transplants. I mean, it's clearly been on our radar screen for a long time. And, there have been various holdups. We all recognize that it's an important population. Most of us have basically resigned to the fact that a placebo control trial is not doable. But I would kind of, I guess in a way, I think, I certainly agree that this is a good time, it seems to me. Opportunities to work with companies who now have perhaps a motivation for getting into this arena and doing a comparative trial. I mean, we can look at the numbers, et cetera, but I think maybe the time is right to do a comparative trial. There are a number of a number of companies out there. As Tom mentioned, the main surveillance study should give us sort of a landscape of what people are really doing. Use that as your standard, if you will, and design a protocol from that. And it doesn't have to be, and probably will not ever pass muster to be a placebo-controlled trial, but a trial comparing a standard, whatever that turns out to be, to drug X, I think, is now really quite feasible. And you know, again, and we have several potential partners to go to. So I'm really enthusiastic about it, and I'd like to see that move forward. Honestly, I think this is the time to strike for that, if you ask me.
So, Bob, thank you for bringing the lung transplant prophylaxis up.
Can you bring the microphone closer?
Okay. Every few years ago, you know, there is a survey going on. The last time was like 5 years ago when, you know, it was noted that about over 95% of lung transplant center receive a prophylaxis. So that is not the problem. The problem is whether people use a universal versus targeted prophylaxis. And, you know, in that review, it was split like a 50-50, 49 and 51%. So if you decide to do the prophylaxis, you know, we have to see like carefully what kind of program people are doing, universal or target prophylaxis. And those programs, I think it depends on the induction therapy. Like for CAMPA, forget it. It has to be universal, but other program, they might just be very happy with targeted prophylaxis. So it's one of the issue that we have to discuss as well.
Yes, sir.
So let me be sure I understand the lung transplant question. Currently, there's not anything indicated for prophylaxis in lung transplant. Is that correct?
Nothing has FDA clearance.
Nothing FDA approved. But it's such a standard of care now that no one would be willing to do a placebo-controlled study. Is that also true?
It is, and I think like Hong mentioned, there have been surveys that have been sent out before, and the majority of lung transplant centers, both in the U.S. and Europe, do prophylax. And now we also have guidelines. The International Society for Heart and Lung Transplant guidelines relatively recently, in the past couple years, were published and do recommend mold-active prophylaxis for the first, I think it is 100 days after lung transplant. And I think actually the IDSA guidelines also recommend anti-mold-active prophylaxis for the first, I don't know exactly what the statement says, but for the first 100 days after lung transplant. So we have guidelines that recommend it. We have centers across U.S. and Europe who are using prophylaxis. I think as Hong was trying to point out, and you elegantly said, clinical practice is a mess because there's nothing approved. The centers are doing their own thing, so to speak, and so some of them are using aerosolized amphetamine. I think that the two most commonly used drugs are aerosolized formulations of amphotericin B and or historically itraconazole, more recently voriconazole. I think some centers may be moving to isavuconazole now that it's on the market, but all of this is without clearance of the drug. I think there are pros and cons to each of those drugs. Clearly this is a complicated population where drug-drug interactions are important.
So the thing this reminds me very much of the design issue we had around drugs for intra-abdominal infections. And if you look at the FDA guidance for intra-ab, there's an appendix about how they estimated the effect size of treating versus not treating using a lot of indirect older data. The antibacterial community has struggled with this problem. You got a disease where no one's willing to use placebo, but the effect size of a new drug versus placebo is kind of buried in the past, and you can't get there anymore because you can't do that study. There have been examples of working that out, because what Pete's pointing at, a comparative trial, intrinsically that's a non-inferiority design, and you're going to have to be able to say, I think that when I don't do anything, the rate of bad news is 70%, but when I do something, the rate of bad news is 20%. You've got to be able to you've got to be able to say that. And I encourage you to look at the FDA guidance, the appendices, not the guidance itself, but the appendix about how they estimated effect size in situations where we didn't have a lot of data. I think you might find some useful correlates there, and it's be a great workshop, but you got to bring your data, be something the FDA be willing to interact with you on, but you have to show up with your data.
So, John, taking that a step farther, and Barbara, trying to find a pathway for a lung transplant, and Pete's correct. We review it every MSG meeting, and it's somewhat just shelved. But John perhaps has opened up a potential doorway, and that is if we look to prior literature, pre-itraconazole, before prophylaxis was being used. There is a body of literature that clearly describes the frequency of fungal and anastomotic infections, as well as invasive pulmonary parenchymal infections. If we were to pull that literature together, pre-itraconazole, in essence, prophylaxis, and present that, could one then emulate the model of intra-abdominal infection and say, here is the anticipated risk if you do not treat, and then go forward with a comparison of what anyone would accept as a standard of care versus a novel agent? John?
All righty. I think it was a productive dialogue, and in the afternoon we will be summarizing all these points to a great degree. Okay.
Go ahead. Well, I want to thank y'all for that was a great discussion. The presentations were terrific. The discussion was really interactive, and the listeners were involved as well. We are going to, I think we can mosey up to, the lunchroom at the Huntley and be back here at 1:30. Just try. We'll try to remain on time so we can be so we can take a good break at 5 o'clock. And looking forward to the afternoon. But thank y'all did a fabulous job, and really appreciate it. Thank you.